I'm Laura Ballweber. I'm an associate professor with the College of Veterinary Medicine and Biomedical Sciences at Colorado State University.
I'm Bert Stromberg. I'm a professor of parasitology at the University of Minnesota College of Veterinary Medicine.
I'm Lou Gasberry. I'm recently retired from the Agricultural Research Service, where I was the research leader for Bovine Functional Genomics Laboratory.
The next phase was the parasite reduction phase. The first step was to look at parasitism and what were the numbers that we had when they treated, and then we asked them to collect samples 7 to 14 days later.
At the present time, the only way to assess potential worm burdens or populations in the host without sacrifice of the host and recovery of the worms is to measure the number of eggs that are being passed in the feces of the cattle. Well, this is not a perfect measurement. It is probably our most precise measurement at this time. So it would be an enumeration of the eggs of an animal at a given time. The idea is that drug treatment or antihelminthic treatment should kill the adult worms that are in the animal, and hence the egg count will drop because there won't be worms passing the eggs. So the standard procedure that is pretty much adopted worldwide is to, as Bert said, to sample a given number of animals, treat those animals, and then sample approximately 2 weeks later. 2 weeks is chosen because longer time period could impart new infection in the host and the reestablishment of new worms. In a shorter time could be misleading because worms potentially could stop laying eggs for a period of time but still be there. So what we did, as Bert explained, is we asked the producers to follow their normal deworming procedure and then 14 days later send us 20 samples from animals, from material that was either directly taken from the animal or to have been observed to have been dropped from the animal. So very fresh material. That went to one of the 3 labs for analysis, and it's important to note that the first sample, the phase 1 and the phase 2, went to the same lab, and they were sent randomly to the labs. We would then compare the egg count in the second sample to the first and express it as a % reduction. In other words, if the starting egg count was 100 eggs per gram and after treatment the egg count was 25 eggs per gram, that would indicate a 75% reduction in the count. This test is used as part of the licensing procedure for drugs, antihelminthics. It's not the only thing, but it's one of them. And normally the average fecal egg reduction for the information submitted for licensing should reach about 90%. So if we take that 100 eggs per gram, we would expect our samples to come in at 10 eggs per gram or less. Our assumption here in our hypothesis, or if not our hypothesis, our use was to see if what the producers were doing on their locations was giving them the expected level of reduction in worm burdens after treatment. Okay? Now, remember, that could involve 2 things. It can say, are they doing things right? And are the drugs they're working with working? So there are multiple things involved here. But The interesting thing to us, I think to all of us, were that fully 1/3 or approximately 1/3 of the operations we looked at didn't reach an 80% reduction in the egg counts, which was surprising at that level. And if we moved up to a more stringent measure, a 90%, almost half of the operations didn't meet that level. And it appeared to us that, or at least if we moved back to that 80% level, all of the operations below 80% had used a pour-on formulation of some kind. The other thing we did then is we tried to get an indication of the parasites that were actually involved in these less than expected level of treatment efficacy. What we tended to see was a large proportion of these were Couperia, the genus of intestinal worms, Couperia. Also, roughly about 2 out of 3 of the drug, I won't say failures, but the treatment, the less than effective treatment actually resulted in changes in the worm populations, which would be indicative perhaps of the appearance of parasites that are more tolerant, worms of a given species more tolerant or more resistant to the drugs. And about 1/3 of them appeared to not have changed the fauna at all, and this would probably be more indicative of a treatment mistake rather than a drug indication.
Question our part, it was evident that only about 5% of all producers do any sort of fecal exams to try and determine what is going on in terms of the parasites in that animal. And they're using primarily appearance of an animal to dictate whether or not they think their program has worked, that their dewormer has worked. So they're not doing any kind of diagnostic follow-up. My personal opinion is that I would like to see the producer and the veterinarian become a team again, and that veterinary input into the diagnosis of parasitism, veterinary input into proper treatment of the parasite issue, I think is a must.